Ladies and gentlemen, on the behalf of the Baltic-German University Liaison Office, I would like to welcome you to our scientific evening, A Night to Remember. And first of all, I would like to introduce you very briefly to our office. And its aim is to act as a scientific bridge between the Baltic states and Germany, to be a platform for scientists and scholars and society to discuss current topics by supporting also scientific projects and organizing scientific events like this event today, which is dedicated to very topical subject, memory and its disorders. Yes, and our ability to remember shapes our experiences, guides our decisions, and preserves the essence of our identities. And in order to discuss this very hot topic, the scientific evening was organized, and I would like to thank our cooperation partners for all the support, the Rotary Club Bochum Rechen and the Vilnius University, especially Ramunė Dirvanskienė. And today's scientific evening will start with the presentation, How Does My Brain Remember? By Associate Professor Ramunė Dirvanskienė, and we will continue with the guest lecture, Memory Traces of Stressful, Traumatic and Forgotten Events by Professor Nikolai Axmacher. And however, before we begin, I would like you to meet our first speaker, and I will introduce her to you very shortly. Dr. Ramunė Dirvanskienė is an associate professor at Vilnius University and an author of a popular neurophysiology book, How the Brain Works. She earned her PhD from the University of Edinburgh and gained clinical experience at the M. Rowling Regenerative Neurology Clinic, working on research projects and clinical trials. Her areas of interest include brain aging, neurodegenerative diseases, and cognitive epidemiology. We are now looking forward to hearing your presentation. Yes, please.
Thank you.
Yes.
I'll just wait for my slides to pop up. So today we have a very mixed audience. I can see we have a lot of psychologists in the building. We have a few neurobiologists. We have some philosophers. We have people from the general public. And we have our guest speaker, Nikolai Axmacher, who will talk about the neuropsychology and the neuroscience of memory. And I know that not everyone is very fluent in the language of neuroscience. And thinking about my introductory 20 minutes, what they could be best used at, I thought that it would be best used just to bring you all to the same page, to bring you all to the same understanding on how our brain remembers. So where is memory in our brain? We know that it's in there somewhere, but where exactly? So that's what I will talk about. So most psychologists think that in our brains, all memories are stored in the hippocampus. We all know that hippocampus is very important for memory, but there's an illusion that hippocampus is a magical box where memories as folders travel and spend the rest of eternity living there. Unfortunately, that's not the case. It's not that easy. And I will go through many important aspects of memory that we have to know about where different types of memory live in our brains. So, of course, hippocampus is very important. It lies in the medial temporal lobe, deep within the brain hemispheres, and it's very important in learning. We have to thank our hippocampus for the ability to learn, especially for the ability to bind different modalities together. When I will remember this event in the future, I will remember your faces, my topic, today's date, and many other aspects, binding them together. And it's thanks to the hippocampus that I will be able to do that. I will be able to bind all of these different perceptual modalities together to form one coherent memory. And then after that binding, after that memorization, each traces of memory travel to their respective locations in the lobe. So long-term memory lives in the cortex. And depending on the modality, its final destination may differ. So visual memories tend to live more at the back of the brain, so colored purple. Motor memories, memories how to perform movements, tend to live in the motor cortices and the associative cortices of the motor lobes. Auditory memories, memories related to hearing, to interpreting sounds, tend to live in the secondary, tertiary associated cortices colored in yellow. And then memories related to body senses, to perceptual abilities, what I sense, tend to live in perception areas, associative areas. You can find them colored in orange. Of course, it's oversimplified because this is a talk for the general public, just to bring you all on the same page. We can simplify it. So depends on the modality of the memory. It can live in the ventral pathway or in the dorsal pathway. So if you call it a semantic memory, what I remember, I'm looking at an object and I remember that this is a pencil. It's a semantic memory. So that memory resides more in the semantic areas of the temporal lobe on the sides of the brain. Whereas memory regarding to location in space, spatial understanding, it resides in the where pathway or the dorsal pathway colored in green. And this is where after processing such memories go and such cognitive processes lie there. Besides the important function of learning and binding memories together, our hippocampus also places the place cells. We have to thank for our hippocampus for our ability to remember spaces. It shows how, because the same one part of the brain is responsible for learning and for remembering the spatial orientation, we know how important orienting in space and attaching it to memory is important is in our species evolution. So we can make hypothesis that our species has developed memory as part of the navigation, where the food is, where the danger is, how to come back to safety. So our memory is very much linked to spatial orientation, and the same hippocampus, besides the functions of learning and binding, it's also very important in it houses the functions of remembering the place. This is why when we go back to the places that we have once visited, suddenly all memories pop up. When you go back to your high school, all the memories of the events that took place there, you start to remember them. Why? Because your place cells activating all the memories that are binded to that place. Memory is also very emotional. So you will hear a lot about that in the following talk, about the links between the amygdala and the hippocampus. But amygdala is the center in our brains responsible for memory, for motivation. And if an event causes us memory, of course, we will remember it much better. And it's not only the amygdala that is linked to emotional memories, it's also the ventromedial prefrontal cortex, because not only we feel when we remember, we also have to evaluate those emotions. Are they appropriate for the social context? We learn emotional behaviors, we compare current events to the past consequences, and we decide either to push the go button on our behavior or the stop button on our behavior based on our memories of the emotional consequences of such events in the past. Memory is also learning. So hippocampus is very important for learning, but so is the frontal cortex. Other regions which are important for the learning and the attention aspect is the dorsolateral prefrontal cortex, colored in purple. Thanks to this part of the brain, we can focus the attention, we can allocate attention towards the objects that we want to memorize, the objects that we want to learn. This is also the place that is very important for our working memory. What objects do we want to learn? Well, we want to learn all about the objects that are linked to rewards. When is it easy for us to concentrate? When the objects of concentration are linked to, when they are deemed significant, when they lead to rewards. And a lot of memories about the rewards, about the significance of events are stored in the orbitofrontal cortex. Memory is not only episodical. We also remember a lot of procedures. We form habits. We not only store knowledge, but we also form habits. And other parts of the brain are involved when we learn procedures. For example, the basal ganglia on the top right. Basal ganglia is involved when we learn voluntary movements, when we learn procedures, when we form habits. And of course, motor cortex, because when we learn movements, we perform them with our limbs, we do motor actions. And the cerebellum is also involved in motor learning and motor execution. So a lot of brain parts are involved in memory. So when we say that memory is all about hippocampus, it's I hope that my presentation has expanded that perception. Memory is not only hippocampus. There's different types of memory. There's working memory, there's semantic memory, there's short-term memory, long-term memory. Different parts of the brain are involved in different important aspects of memory. Now, going back to my own research interests. So my interests are cognitive aging and what happens to our brain as we get older. So our brains, they change. There's a loss of volumes, cortical thinning, loss of white matter in certain brain areas. And we all know that with age, we lose some of our memory abilities. It can become a bit more difficult to memorize. As older people tend to complain of memory problems. So how are neuroanatomical changes related to these memory problems? So these are the questions that interest me. And when we look at studies of cognitive aging, we find that this volume loss tends to be mostly it starts with the hippocampus and the frontal lobes, and these are the areas that show signs of shrinkage first, and they are most clearly exhibited. However, the temporal lobes, they don't seem to lose volume with age, and when we observe structural changes related to brain aging, we don't see evidently brain matter shrinkage in these regions. How does that relate to brain functions? How do different functions of memory change when we get older? So we see that with age, there's a decline of speed of processing, there's a decline of working memory, of long-term memory. However, our semantic memory, word knowledge memory, does not decline with age. It even tends to get better. Why? Because these functions are stored in the temporal lobes that I have shown in the picture before, and this area of the brain does not seem to lose that much volume with age. And that's why when we get older, our semantic knowledge remains the same, either gets better. When people ask me if I want to keep my brain healthy in older age, should I do crossword puzzles? No, you shouldn't, because with age, semantic knowledge, word knowledge does not decline. There's no purpose in doing crossword puzzles. If you want to keep your brain healthy, you should do activities related to information processing speed, working memory, learning, especially if that's fast-paced processes. Why do older people enjoy crossword puzzles so much? It's because they're so good at it. So there are individual differences in how our brains age. For some people, these processes are faster, for some are slower. And it's one of the research questions that interests me a lot. So what makes some people's brains more resilient than others? I've included a model of resistance to cognitive decline proposed by Denise Park and her colleagues, just as an illustration of the theoretical models going on in the area. But to sum up, some people's brains, they have better brain reserve. They just have better working brains, more robust brains. Some people's brains have better brain maintenance, so they are much better to cope with the processes that start to happen in their brain. And this brain maintenance is hugely impacted by the lifestyle, by our lifestyle choices, by the substances we use, by how active we are physically or mentally. And then the third factor that causes individual differences is cognitive reserve. And that's the area that is particularly interesting to me. How can we use our existing cognitive skills to compensate for the newly happening damage? And in cognitive literature, this is called the cognitive scaffolding theory. Scaffolding is a term that comes from construction. When a building starts to fall down, how can we help it stay up? And we put scaffolds around that building so that it wouldn't fall. And cognitive theories propose that we can employ some of our cognitive functions to compensate for the loss that we experience with aging. And theories of cognitive epidemiology, why some people are more resistant to age-related memory decline, propose that it is because of three reasons. It's because of brain reserve, brain maintenance, and cognitive reserve and cognitive scaffolding. So thank you for listening to me. I hope that my brief introduction into the area of memory and neuropsychology has brought up some of you to the same level that others have. I know that some of you who came here today have more knowledge about neuropsychology, and I hope that my talk will help you now better understand our keynote presenters' research. So the stage is yours, unless we have any questions. No questions, so the stage is all yours.
No questions in the moment, or some comments? Okay, yes, please.
Thank you for the great presentation. So I actually have a question. So why do you think is the lateral temporal cortex so resistant to age?
I hope you will answer this question. I can only observe individual differences, tendencies. Maybe it's because semantics are more what we were talking before we came here. It's more about associations, about that fifth or sixth level of cortex. I don't know. It's a good area for studying.
Yeah, we should find out.
We should find out.
No more questions in the moment? Yes, then I would like to thank you for your presentation. Thank you very much. And now let us now continue our scientific evening with the guest lecturer by Professor Axmacher. He is the head of the Department of Neurophysiology at the Institute of Cognitive Neuroscience, Faculty of Psychology at the Ruhr University Bochum, and he combines functional MRI at 3D and 7D and intracranial EEG recordings to investigate the neural mechanisms of memory functions and their dysfunctions related to Alzheimer's disease and post-traumatic stress disorder. This work has been published in more than 140 articles, including papers in Science, Nature, Neuroscience, and Neuron. and he was awarded several prestigious grants, most recently a European Research Council Consolidator grant in 2019. Thank you very much for coming here to Vilnius and joining us today. The floor is yours, please.
Thank you for the nice introduction and for inviting me to come to Vilnius. It's a wonderful opportunity to meet you and to be at this beautiful place here. So as you can see, my university in Bochum is not as old. It's also not as beautiful. It's very big, but it's a good place for research as well, I would say. And this is a very good introduction when it comes to spatial memory, because if you imagine being at this campus here, you need at least I would say like 2 or 3 weeks to learn your ways. It took me 4 weeks because I'm very bad at spatial navigation. It's a very complex place here, but once you know your way, it's beautiful because you can very easily communicate and collaborate with your colleagues from other faculties. So as mentioned before, the title of my presentation today is about memory traces of stressful, traumatic, and somewhat paradoxically forgotten events. Indeed, I believe that it follows nicely up on the previous presentation by Dr. Dirvanskienė. I will not talk that much about the hippocampus. I will more talk about the different contributions of neocortical areas as well as the amygdala for memory. Can you hear me well, actually? Okay. If you have any question, please feel free to interrupt me at any time. Otherwise, I'm also happy to answer them afterwards or during water and juice. So traumatic memories, intrusions or flashbacks are the core feature of post-traumatic stress disorder, but also of many other psychiatric diseases and in particular anxiety disorders. And in my talk, I will not only try to address some basic questions related to what memories are and how we should conceptualize memory traces, how we can investigate memory traces, but also some more applied questions related to pathological memory traces. So how can we understand traumatic memory traces? What makes traumatic memory traces different from normal memory traces? And how, at least from a conceptual point of view, can or should therapies try to improve traumatic memories? So I'm not a clinician. I cannot give you like a new therapeutic option, but I would like to share some conceptual ideas how we should understand the changes that are brought about by psychotherapy or also other kinds of interventions to pathological memories. So how can we describe the specific features of traumatic memories as compared to healthy episodic memories? On the one hand, it has often been described that traumatic memories are or maybe limited to a central event. And this has been described as the weapon focus effect. Who of you has heard of the weapon focus effect before? Okay, so just imagine you're taking a bus to this university as every morning maybe. Then usually you don't remember that much from this episode, but suddenly someone pulls a gun and points it at you. So first of all, you're much more likely to remember this particular morning as compared to the other mornings, but you may do so very selectively. You will very likely remember that there was someone standing with a gun. You may also very likely remember particular features from this person. On the other hand, some of the more peripheral aspect of the scene, so some of the other passengers or how the bus looked like, may be forgotten. So it is a very selective aspect of traumatic events that is very vividly remembered, and actually more vividly remembered than normal episodic memories of everyday events. But some of the more peripheral, less relevant details are less well remembered. This is not only the case for very like for real traumatic events. There's also been described for events like more everyday events that cause a certain degree of psychosocial stress. So if you are in an exam situation, you're again very likely to remember how your examiners look like, some of the maybe what they were doing during the exam, but at the expense of some of the peripheral details. Traumatic memories have also been described to be more perceptually detailed and less semantically elaborated representations. So what does that mean? On the one hand, trauma survivors often describe that when they re-experience a trauma in the, as an intrusive memory or even as an almost hallucinatory flashback, they this has very strong sensory vividness. So they experience both the visual characteristics, but also other sensory characteristics of this event. So traumatic memories are very sensory. On the other hand, they are often less semantically elaborate. So it is more difficult for a person to describe the entire meaning of this event, and also, here we come to the next point, to integrate it with other events. So traumatic memories are often described as distinct from other experiences in your life. And this is because traumatic memories, by definition, are unexpected and violate the expectations that you have. So just remember from your bus ride, the person pointing at the gun with you is in particular traumatic because you're not experiencing that every day, because it's something that you don't have any expectations or predictions for. Therefore, you cannot integrate this experience with the other predictions and the stories that you tell about your life, so your life narrative. Finally, the traumatic memories have been described as being overgeneralized. So this means that they can be easily triggered by ubiquitous sensory cues. So again, trauma survivors describe that they are often haunted by the recurrence of intrusive memories or intrusions, and they don't really know what triggered these events. But they can occur in a multitude of different situations. And of course, there are some triggers, but they can be very remotely related to what actually happened. And these four features here are some of those for which, from a cognitive neuroscience perspective and from the perspective of memory traces, I will try to give some clues on how to understand them. So first of all, one could conclude that traumatic memories are, on the one hand, characterized by specific content. So what you remember, so the traumatic episode that forms the content of the traumatic memory, of the intrusion or the flashback. But also they are characterized by specific experiential qualities. So it's not only that what you remember is different, it's traumatic in this case, but you also remember it differently. So how can cognitive neuroscience research contribute to an understanding of traumatic memories and of their possible transformations due to therapeutic interventions? So the main ideas that our research is building on are that traumatic memories depend on specific representations in the mind, and then, of course, also in the brain. These representations have, as mentioned before, both a specific content, what is remembered, and a specific, what I would like to call, representational format. And I hope that this term will become a little bit clearer, which described how a particular event is being remembered. And therapies, I would like to suggest, should not only try to change the content of what is remembered, because this is something that has appeared before, and I would suggest that psychotherapies or other kinds of interventions do not want to induce false memories. They do not want to change the content of what is remembered, because it has happened in the past. But they want to modify how this memory is remembered, how this content is remembered. So we need a conceptual and empirical framework that allows to track the neural representations of specific contents. So to identify memory traces, or as it has also been called, engrams. We need also a framework that allows to identify these different representational formats of the same content, and that allows measuring the transformations of these formats. So how can we measure engrams? If you have rodent researchers, we can do that relatively directly. So we can use optogenetic or genetically modified rodents that in a particular situation, which in these rodents is a kind of a stressful or even traumatic situation when it receives electric shocks, where in this situation we can label all those cells that express certain kinds of genes which are associated with plasticity and learning. And then afterwards, we can use optogenetics, so light fibers implanted in the brains of these rodents, to again activate all of these cells and bring back the memory to this particular event. And there have been many studies using these kinds of manipulations. They've shown that it is possible to even modify afterwards these memory traces, and that also some memory traces that for the rodents have not been accessible for a certain period can be brought back. So it's a very powerful methodology. In humans, obviously, this is not applicable because we don't have the luckily not the genetic manipulations available. We don't want to implant light fibers just to do research. So all of these methods here are not feasible. On the other hand, there have been in the last maybe 10, 15 years, developments of machine learning methods to analyze neuroimaging data from EEG, scalp EEG, but also from functional MRI that allows to identify specific contents. And these methods are more or less complex, but here's basically the gist of them. So just imagine that you're perceiving two different stimuli. So you're perceiving the stimulus of a hand, and you're perceiving the stimulus of an umbrella. Now, the hand and the umbrella, like from a perceptual perspective, look very different. So in your sensory areas, the representations of the hand and of the umbrella is very different. And these representations, so the activity patterns in sensory areas elicited by the stimuli, are schematically depicted here. So this is basically the pattern of activity in a certain brain region measured with EEG or fMRI, and you can compute the similarity or dissimilarity between these two stimuli. So in sensory areas, they would be very different. On the other hand, you could assume that in the premotor cortex, where action plans are represented, these representations are strongly overlapping. So, so basically, you can use these methods to identify the neural activity pattern that is specific for one stimulus, so that is different between the hand and the umbrella. And you could also then look at the pairwise similarities of these pairs of stimuli and compare them between different brain areas. And depending on what is processed on a specific area, these patterns are more or less similar or different from each other. So how can we now use these methods to study traumatic memory? So on the one hand, one could think, okay, the actual traumatic memories are occurring in patients with post-traumatic stress disorder or other kinds of psychiatric diseases. So would it be possible to study these patients while they're lying in a scanner, in an fMRI scanner, for example, and wait for intrusions to occur? Right? I mean, this would be the most naturalistic and ecologically valid approach, but obviously this is very difficult. It may take for a long time before these intrusions occur naturally, and trying to trigger them is ethically dubious, I would say, because the patient is then in a vulnerable environment inside this dark and loud scanner, and trying to induce intrusive trauma-related memories in a distressed patient is very problematic. So yeah, this kind of research cannot really be carried out for practical and ethical reasons. On the other hand, of course, the encoding and then the actual experiencing of traumatic events cannot and should not be directly studied in healthy people, right? So we cannot invite our students and traumatize them. On the other hand, what we could use as cognitive neuroscience researchers is try to use preclinical models that try to mimic certain aspects of intrusive memories at a level that is not distressing and does not cause any lasting problems in our study populations. So the most commonly used and most widely known paradigm for that is fear conditioning, as you know, right? So as like probably the most widely known approach to study fear learning. In some cases, it's even used to study episodic memory. I'm not really sure whether this is justified, but yeah, I'm happy to discuss that afterwards. Another method would be to show negative images. So maybe you know the IEPS data set. So this is a data set that has been collected some decades ago with the gory pictures of accidents and some disgusting scenes. There's also some more positive and some neutral images. You can show them to your participants and study their reactions. This is, on the other hand, a very both of these methods, I would say, in humans have their problems because fear conditioning experiments in many cases induce one-trial learning and do not induce any of the intrusive memories that occur in traumatic conditions. The same is true usually for the IEPS picture set. So what we are using here, or what we are focusing on, are two paradigms that are a little bit more ecologically valid while still being ethically unproblematic, I would argue. So one is a psychosocial stress induction paradigm. I will show that in the next slides what we did. And then finally a trauma film paradigm in which we show our participants trauma analog movies, which are the kind of movies that people pay to watch in the cinema. So both of these conditions here do not induce measures of distress that are uncommon in everyday life. So the psychosocial stress induction paradigm uses a paradigm which is called the Trier social stress test because it was developed in Trier and capitalizes on social stress. So participants are invited there. Have you heard of the Trier social stress test before? No? Okay. So participants are invited to come to an experiment, and then they are given the task to prepare for a fictitious job interview. So they are asked to imagine their dream job they would like to obtain, and then like think of some arguments to convince an audience that they are the optimal candidate for this position. And then after a few minutes, they are invited to stand in front of an audience and to present themselves. They do not know how long they have time to present themselves. They are not given this information. They are just given the information that the audience is listening to them, is evaluating them, and that they're being filmed at this time. So I've participated in this experiment myself, and I can tell you it's stressful. Even though you know it's completely fictitious, so you know that there's going to be no consequence, you feel stressed yourself. You may feel angry. You may feel ashamed. You may feel unhappy about the situation. You can also measure that the cortisol level increases in this situation. But then on the other hand, it's not anything that is actually traumatic, and it's somewhat comparable to exam situations, with the difference that in an exam actually there is a consequence for later life. So unbeknownst, there's two factors here that the participants are not informed about. One is that the audience, who is consisting of fellow students, is instructed to only give negative and reserved feedback. So different from exam situations, at least as they should be conducted, the examiners, or here the audience, is not approvingly nodding here and there or smiling, but actually they remain a stern and reserved attitude. The second aspect that in this version of the Trier social stress test the participants are not informed about is that the things that are lying around here on the table of the audience, in front of the audience, actually play a major role in the experiment. So there's a number of different things here. So for example, there's a teacup, there's this Coke can, there's like a book, a pencil, and so on. And some of these objects are used by the experimenters during this experiment. So for example, the experimenter would write down some notes with a pen on a sheet, or would use the tea cup here to pour in and drink some tea. On the other hand, there are some other objects which we call peripheral objects, which are just standing around on the table and are not manipulated by the experimenters here. I should also mention that some of the students who are invited to the participants are in the lucky situation that they are recruited randomly into the control group. So they are not participating in this nasty job interview here, but they are just asked to tell some holiday experiences or talk about their hobbies to the experimenters. And in this case, also the experimenters give some friendly feedback and actually talk to the students. And what you can see is this is not subjectively stressful and there's no increase in cortisol levels. So it's a good control situation. On the other hand, the situation is very similar. There's the same pot of tea standing around that the experimenters use according to a specific script. There's the Coke can and so on. So what has been shown before now a few times by the group of a collaborator of mine, Oliver Wolf, outside Bochum University, is that in this stressful group, so the actual Trier social stress test, the memory for the objects that are manipulated by the committee is specifically increased. So this is what is shown here. So here's the memory for both the central and the peripheral objects. The red bars are referring to the stress group, and the green bars to the friendly control group. And here you see that both memories are actually increased in both groups. In other versions of the experiment, only the memory for the central objects is increased, but also in our version, there's a significant interaction in the memory such that it's disproportionately increased for the central as compared to the peripheral object. So this is somewhat modeling this weapon focus effect, that in particular the central events of a stressful, of a distressing event are better remembered afterwards. So now we combined this experiment here with an fMRI study, where after this day, when the participants were invited to the audience, they were coming back to the scanner and were in the scanner presented pictures of all the central and peripheral objects as well as neutral objects. And we used the analysis approach that I just showed to look at the similarity between the central objects, between all central objects, between all peripheral objects, and also between the central objects as compared to the new objects. And we tested different hypotheses, how the similarities actually could be related to memory. So on the one hand, we thought, well, maybe the central objects are better remembered because their representations is very distinct. So let's say the teapot has a neural pattern that is very different from the representation of the cup or of the pencil. So all of these memories are well distinct, and therefore they are not confused with novel events. On the other hand, one could also assume the opposite. So one could assume that the memory is better for the teapot and the cup and the pencil that the committee is using, because they are all very similar to each other. So they are bound together into one coherent episode of this trauma, not trauma, but this stress-related episode here. So we had these two hypotheses, and we analyzed the activity patterns to the pictures of all of these different objects in the scanner in these two brain areas here that have been mentioned before, the hippocampus and the amygdala. And here's what we found. So we found that actually the second hypothesis was supported. So we found that the similarity patterns between the central objects in the amygdala was much more similar to each other, as compared to the representations of the peripheral objects, or as compared to the representations of the central objects in the friendly control condition. We did not only show in the scanner pictures of the individual objects, but we also showed pictures of the experimenters. So this was more of an exploratory addition to the experiment. But we were interested to see whether maybe these pictures of the experimenters, even though they're obviously perceptually very different from the teapot and the other objects, maybe they also become more similar to some of the objects. And indeed, we found that this was the case. Again, in the amygdala, the representations of central objects became more similar to the representations of the faces. And the more this was the case, the better these objects were remembered. So those objects whose representations in the amygdala became more similar to the representations of the faces were afterwards particularly well remembered, suggesting that this is not just a fancy and complex analysis method that you could apply, but actually it explains some of the behavior in this experiment here. So this is just a cartoonish drawing again. So we found that in what we would like to call the representational space in the left amygdala, the n-gram patterns, or the neural representations of central objects, are bound with the representations of the stressor's face into one generalized memory of the stressful episode. Okay, so to summarize this part, we found some evidence here that for the weapon focus effect, namely that there was indeed something specific in the neural representations of the central events as compared to the peripheral events. And what is specific is that they are represented more similar to each other in the amygdala specifically. We didn't find that in the hippocampus. So the second study, which I would like to present, has been done by my PhD student, Malte Kobelt, and a postdoc, Gerd Waldhauser, from my group. There's no reference here because it's currently under revision, so that's all fresh. But yeah, hopefully it will be accepted in the near future. And in this experiment, we did not use a stress intervention, but what is called the trauma film paradigm. So this is the arguably most established paradigm to study intrusive memories in healthy participants. So what we did, and what many other researchers did before, is to find a number of short movie clips of about 30 to 60-second duration of aversive or a trauma analog events. So the kind of events that if you experience them in real life, either yourself or if you witness them in real life, have similar contents to what is to traumatic episodes. So I spare you all the details, but so there's some clips about animals, some clips about interpersonal violence. Maybe some of you have seen, just to give one example, this movie of a person who was hiking in the desert and got trapped and couldn't pull out his arm. So the only way for him to get out of the situation is to take his relatively blunt knife and cut off his arm. And this is very graphically depicted in this movie. And we find a nice 40-second scene focusing on this event here. And I watched that, and I had some intrusive memories of that for quite a while. So this is an established paradigm in research since many decades because it does induce involuntarily occurring intrusive memories. It's very important to select a healthy control population. So you cannot do, I would argue, you cannot apply this paradigm in psychiatric populations, but only in healthy, very carefully screened students. And then what happens is that you induce intrusive memories that occur with a particular frequency. So you can ask the participants to write them down in a diary, but they disappear automatically spontaneously after 1 or 2 weeks. So we have now conducted similar paradigms in probably like 250 students, and none of them had any complaints after a while. But I think this is because we used strict exclusion criteria to select the study population. We did not only show the movies of these aversive episodes, but we also selected movies of thematically matched control episodes. So, for example, we showed another 40-second clip of someone receiving an arm massage. So the content is roughly the same, right? The arm plays a central role there, but in the arm massage, there's nothing aversive. Or we showed a scene of someone driving along a highway. And these 21 trauma analog clips were randomly intermixed with the 21 control movies. Then we presented that in the scanner. And this was followed by a 10-minute period of resting state where we asked the participants to indicate with a button press whether they witnessed an intrusive memory. And if they pressed a button, we asked the patient with an MR-compatible microphone while the scanning was ongoing, which particular event they had just witnessed. So, for example, they would press a button and they would say, well, I remember the person with a knife in his arm. We had a number of participants who did not experience any intrusive memories in the scanner, but we also had a substantial number of participants who experienced some intrusive events here. So up to 22 here, I think is the maximum. And we also had some involuntary memories of the control movies, but significantly less. So now we used again the same analysis approach as in the previous study. So we looked at the activity patterns during the presentation of these 40-second or 30 or 60-second movie clips and compared then the pattern of activity either between different trauma movies. So this is shown here, between pairs of these trauma analog movies, or between pairs of control movies, neutral movies. So this is called NN here, neutral, neutral, or between one trauma movie and one of the neutral movies. And we selected only those pairs that did not have the same content. So not the arm with the knife and the arm with the massage, but any of the others. And we tested whether there were some brain areas where the trauma movies were more similar to each other than neutral movies were to each other. So where the trauma movies were more their representations were more similar. And we also tested whether the opposite was true in other areas, so that maybe the neutral movies were more similar to each other than the trauma movies were. And we tested whether there were some areas where one trauma movie had a representation that was more similar to any of the neutral movies than the neutral movies among themselves. And this is what we found here. So in the first contrast, we see in red those areas where the trauma movies are more similar to each other than the neutral movies. And you see that in particular in these sensory areas, representations of different trauma movies were more strongly generalized as compared to representations of different neutral movies. And this, I think, is an even more interesting comparison, because we found that, again, in similar sensory areas here, when a participant watched a trauma analog movie, the representations was more similar to a neutral movie than different neutral movies were to each other. And I think this could be an explanation for why intrusive memories occur. Because if you store a traumatic memory such that it is very similar to thematically completely different neutral memories, this could explain why ubiquitously occurring triggers, sensory triggers, can elicit a memory of these intrusive events. On the other hand, we also found in some areas the opposite effect, so that different control movies, different neutral movies were more similar to each other, and different traumatic movies were more dissimilar. And these were in particular those areas that are relevant for semantic processing. So in the temporal lobe that survives long in age for whatever reason. But in these areas here, the different neutral movies are more similar to each other. So they are putatively more strongly, can be better bound into one integrated conceptual narrative, whereas each of the traumatic movies on a conceptual or narrative or semantic level is very distinct from each of the other traumatic representations. Okay, so this is the second last slide from this study here. So here we also looked at the activity and the similarities during the spontaneous occurrence of intrusive memories. So here the participants did not watch the movies, but just reported with a button press that they experienced the movie, an intrusive memory. And again, we see why they experience an intrusive event, they again have a higher activity in these sensory areas, and also the similarity between different intrusive events is higher as compared to baseline activity, as compared to during baseline. So we found that not only during the experiencing of these trauma analog events, but also during the occurrence as a memory intrusion, there's this pattern of generalized visual representations. So this is again a schematic depiction of the results. So we found that for the neutral episodes, there were relatively distinct sensory representations, and for the different traumatic representations, they became overgeneralized in these sensory areas, and this may explain why they could be triggered also by other kinds of events. On the other hand, the conceptual or narrative level representations of the traumatic events remain distinct and different from each other, whereas the different neutral events can be more easily integrated into one overlapping narrative here. Okay, so we have found some evidence for the more perceptually detailed and less semantically elaborated representations for why trauma experiences cannot be integrated into a continuous life narrative, because they rely on very distinct and separate conceptual representations, and also some evidence for this overgeneralization of traumatic memories. So this is again a brief summary here. So memory intrusions, if they rely on these overgeneralized sensory representations, this is something that may be addressed by therapeutic interventions. So how should these transformation processes be conceptualized, and what happens to representational formats, so kinds of representations that are not desired, that we want to transform? So how do therapies work? Like a very simplified idea is depicted here. I'm not sure whether some of you have seen this movie here. It's called, what's it called again? Thank you. So you've seen that before. Yeah, it's a wonderful movie. If you haven't seen it, and if you're interested in memory, I can highly recommend it. So in this movie, the character here has a difficult, some conflicts with his girlfriend, and actually wants to separate from her, and in order to overcome this experience, finds this fictitious company here that offers an apparatus that looks a little bit like a portable MRI, actually, that is in the movie able to zap away individual memories. So basically to delete them. The problem is that during the course of the treatment, after some sessions, the protagonist here decides that he doesn't want to like zap it away, and he wants to have it back, but now parts of the memories are gone, and yeah, this is of course a problematic situation. I can really recommend watching the movie. It's a great film. So is this how therapy works, or is this how therapy should work? So this would be a kind of a transformation that is parsimoniously, or that is like conceptually the most parsimonious, the most simple, where memory transformations are due to a quantitative reduction of the strength of a memory to the degree that if this like strength is completely reduced, then the memory is gone. So this would be a memory transformation as erasure or like removal, if you want. An engram is weakened or deleted. So this is what is commonly referred to when we speak about forgetting. And I have an animation here. So the memory trace in this, according to this view here, would be gone. And this is an irreversible process. It also doesn't come back. On the other hand, a memory trace could be modified by temporarily suppressing it. So this is something that is believed to occur during voluntary memory suppression. I will come back to that a little bit later, or also during processes of extinction learning. So the psychologists here among you probably all know that during extinction learning and also some extinction learning based psychotherapies, memories are not deleted, but they are overwritten or suppressed by other competing novel memories. And it is a common observation that in a different context, so for example, when the patient comes back from the therapist's office and has learned to cope with his spider phobia, then he comes back home, goes to the basement, sees a spider and freaks out again. So basically the fearful memory of the spider is still there. It's just inhibited or suppressed by a competing memory during the extinction learning. So basically the idea is that due to activation of some other inhibitory areas, in particular in prefrontal cortex, so we've already learned in the previous presentation that both the lateral prefrontal cortex but also the medial prefrontal cortex are relevant for that. These memories here are suppressed, but this is a reversible process that requires an active effort, strongly context specific, and if the inhibition is gone, then the memory trace can come back. So is this how therapies should work? That they should like temporarily suppress or reduce the strength of a memory trace, but then the problem is that it may come back. Like another common concept is that during therapies, as well as during other processes such as memory consolidation, reconsolidation, etc., a memory trace is actually transformed. So the engram, the content of the engram is modified. Some information is added, some information is subtracted, and some information is changed. So this is schematically shown here. So you see that there's some new blue pieces of information in the memory trace, and some of the previously existing ones have gone. But now there's in a way, both an empirical and a philosophical question, and is this still the same memory trace? So how much would a memory trace need to be altered in order to be still the memory trace of the same event, right? You can say in practice, I don't really care as long as the patient can live with that, but I think it's still a valid question. So when we modify our memories, is this still a memory of the original event, of the new event? And like how much change would need to happen for the memory trace for it to remain the same? You could also ask, and this has been discussed also in the past, whether this is still my memory. So if you modify the memory, for example, due to psychotherapy, is this something that is a memory of the patient, or is it actually something that the memory is that the patient has come to believe? And there's descriptions of cases where the patients have come to believe situations that actually haven't occurred, so that basically some false memories have been induced in these therapies. You could also even ask, is this still a memory? So is this still something that is actually about a particular episode that has occurred in the past, or is it something that is just afterwards made up? And again, there's some views which say that doesn't matter as long as the patient can live with that. But as soon as we come to legal questions, so did this actually happen? this is actually relevant. And in many cases, I would also say that we don't only want to believe that we can like come up with a pleasant story of our life. We actually want to believe that what we remember has actually happened. So we have so it's actually relevant for us whether our memories are veridical or not. So I would like to suggest a somewhat conceptually alternative framework. I'm not saying that there are no cases of erasure, memory suppression, or transformation, but I would say that some of the phenomena that are often described by these previous frameworks may be better conceptualized by this alternative framework here. And this I would like to call the framework of deep engrams. This assumes that individual experiences are represented by engram patterns with multiple layers. So one memory trace can consist of multiple different layers or formats. These layers correspond to sensory processing steps, so as the levels of processing theory has been described. And we are also assuming that the layers can be measured by the different layers of deep neural networks. I'm not going into that today. We did a couple of studies where we applied deep neural networks to look at the different perceptual or conceptual formats of memories, but that's like for time reasons, I'm not going into these studies. So how should we understand these different layers? So the idea is that one and the same content is stored in a memory trace with different formats. So just think of the event tonight. You're sitting here in this room, and afterwards you can remember this one single content, this one episode on different levels. So either you can try to focus on particular perceptual events. So for example, the color of my pullover, or the shape of the tables, or other perceptual details. You could also try to focus on particular conceptual or semantic events, which could be related to, for example, to the presentation, could also be other conceptual aspects here. And I would also suggest that there's aspects in between. So for example, certain surfaces or body parts or so, which are on the way between a very low level sensory representation and a more conceptual representation. So there's these different representational formats of one and the same memory trace about one particular event. There's also different neural formats. So it's known that these perceptual as compared to semantic or conceptual representations are processed in different brain areas. For example, along the ventral visual stream, we've already heard about the what stream and the where stream, and the what stream also consists of multiple different processing layers. And there's different phenomenological formats. So there's the particular degree of vividness of a memory, emotionality of a memory, visual perspective of a memory, and so on. And these different formats may or may not, in principle, be related. So again, the idea is that episodic memory is could be conceptualized then as a selective recall of deep engram patterns. So when you remember this episode here of tonight, you can, to a certain degree, deliberately focus on a particular layer, on a particular format of this memory. It's relatively flexible. On the other hand, there's some suggestions which we haven't tested yet, but we would also like to do that for stressful or traumatic memories, these formats are more restricted. So trauma survivors describe that when they remember an episode, they cannot to the same degree deliberately focus on particular events, but it's always the same movie that is going on in their mind. So apparent memory transformation then may be in this framework here conceptualized as the selection of different layers. So it's not that the memory trace itself changes when initially you thought about perceptual details of an event and afterwards you focus on conceptual details, but you're actually pulling out or specifically activating specific formats of the same deep memory trace. So also to a certain degree, I would argue, solve these philosophical problems here. So how can we test this idea? How much time do I have left? A little bit more? Okay, yeah, perfect. So I would like to present you two studies, no more fMRI, one behavioral study and another intracranial EEG study where we like try to address specific aspects of this framework. So in the first study, we tested the idea that memory transformations brought about by a conflict between episodic memories and our self-image lead to a transformation or lead to a particular representational format of a memory that could be measured on a phenomenological level. So this may be a little bit cryptic, but so the idea is that this is not our idea. This has also been described by Martin Conway, for example, that autobiographical memory has two main constituents. One constituent is the pure memory part of that, so episodic memories, memory traces, or semantic memory traces. The other aspect of autobiographical memory is the auto part of the word, of the term. So basically the self models, the view of ourself. And what Conway and others have described is how autobiographical memories lead to the build-up of this view of ourselves, so of the self model. On the other hand, we here focused on the reverse direction. So which influence a self model may take on how we remember particular experiences. And in particular, we focused on some events that were inconsistent with how we would like to see ourselves. And these are events that elicited feelings of shame or guilt, right? So the self model has specific aspects, an ideal self, a descriptive self, an odd self. So how we would like to be, that's the ideal self. How we believe others expect us to be, that's the social, the odd self. And how we think we are, that's a descriptive self. And in particular, this ideal self and the odd self are those that are in conflict with memories for those memories that we remember with feelings of shame or guilt. And one could now think that when we remember events for which we feel feelings of shame or guilt because of this conflict, we could try to delete them, or we could try to suppress them. We could try to transform them and modify certain parts so that they become better congruent with what we remember. But it could also be that we just remember them differently, so that the representational format is actually changing. And that's what we tested here. So this was a typical COVID study. So it was actually planned to be done as a lab study, but then 2020 came and Thomas Lukaszewski, here my psychology student, as all of us stayed at home and had to do an online study. So actually what we did was not invite participants to our lab, but ask participants to collect memories of everyday events, and each evening sent us, sent to an anonymized database, sent a brief description of one event of that day that either elicited a feeling of shame, or if there was no event that elicited a feeling of shame, that was an event that elicited, we asked them to think of a feeling that they remembered with guilt, or that was otherwise negative, or that was sufficiently unique to be remembered but neutral. So some examples here, just to give some examples. As I mentioned, this happened during the COVID time. So shameful memories were those where participants said they didn't wear the mask properly in a given situation and felt bad about that afterwards. But also because many of the participants here were students. These were events where they said, well, they went to an online lecture at that time. They were not well prepared. They went to an exam, they didn't perform well because they didn't learn it well enough. So basically their ideal self of how to behave was violated. There were the guilt-related experiences. These were more like specific transgressions. So where they behaved in a certain action different from what they thought others expected them to be. So, for example, they broke something in a supermarket or at a friend's place. So they felt bad about this particular thing, but they didn't think, okay, they were a wrong person because they of a particular episode. A negative event was, for example, when the bike was stolen or so, right? I mean, this is very frustrating, but you wouldn't feel shameful or guilt about that most of the time. A neutral event was a participant buys a new washing machine, right? I mean, this is necessary maybe, but it's neither particularly exciting nor most cases nor negative, but it's also sufficiently unique because you don't do that every day. Overall, we included 65 participants and asked them across a longer period here of up to 11 weeks to send us these descriptions of episodes. So overall, we had more than 3,000 autobiographical events, more than 400 related to shame, more than 500 of guilt, and so on. And for each of the memories now, we asked them about specific phenomenological details. So in particular, whether they remembered the episode from a first person or from a third person perspective. So it's in autobiographical memory research, it's known that while you experience, of course, all your life, apart from like some very extraordinary events, you always experiences from the first person perspective. When you remember a particular episode, you may most often again do that from the first person perspective, but in some cases you basically see yourself as part of this episode. This is particularly described for, again, traumatic events, but also in general negative events and events that occur to a period of your life that you want to distance yourself from. And it's something that occurs more often for events that you've thought about more often. So there's like various aspects that contribute to this third person perspective. It's a particular phenomenological format of a memory. And that's something that we also tested here. We also tested the amount of affective distress that a person reported about one episode and collected all of this here. All of the episodes, all of the more than 3,000 episodes then were also rated for levels of shame, guilt, negative affect, visual perspective, and also centrality. I won't go into that. So what we found was that, first of all, where's it shown? The amount of negative affect was the highest for the shame episodes as compared to all the others. So in everyday life experiences, those events that participants felt shame about were more negative than the negative episodes or the guilt-related episodes, and of course the neutral episodes. We also found that the visual perspective was most strongest in the third person perspective for the shame episodes as compared to the others. Well, this is some, let's skip that. Then what we did was to feed all these 3,000 episodes into Google Sentence Encoder and looked at the semantic similarity between every description to all shame episodes, to all guilt episodes, all negative episodes, and all neutral episodes. And this is the summary slide here. So we found that those episodes that resembled on a semantic level the shame episodes strongest were most often remembered from a third person perspective, and those that received the highest shame ratings. So this shows that self-incongruent memories that elicited feelings of shame were remembered in a particular representational format on a phenomenological level. We're currently doing a study where we test this in the scanner again. So where participants then are asked to remember particular shame, guilt-related neutral and negative episodes in the scanner, so that we can also look at the representational format at the neural level. So in the very last study, which I would like to show you today, we looked at memory traces of forgotten events. So this as I mentioned, initially may sound a little bit paradoxically. So what we did here was a study using the directed forgetting paradigm. I'm not sure whether some of you may be familiar with that. So that's a study of voluntary memory suppression. So in this experiment, participants see individual items. In this case, it's just words. And then after seeing each item, are asked to either try to encode these items like intentionally, or to suppress it and not encode it into memory, right? And even though this may sound paradoxically, it's a very strong, it's a very robust behavioral paradigm. So if afterwards you show all of the words again, so the to be forgotten words and the to be remembered words, the memory of the to be remembered words is much better than the memory of the to be forgotten words. This is a study that another postdoc, Sanatin Över, conducted, and she did not use functional MRI for this study, but intracranial EEG recordings. So this is a bit of an unusual method in cognitive neuroscience. So this is a method that is not done in healthy participants, but in epilepsy patients who are implanted with electrodes to identify their seizure onset zone. So basically, these patients have a pharmacorefractory epilepsy. They come to an epilepsy surgery department to resect a part of their brain causing the epilepsy. And in some cases, in order to identify which part of the brain is causing the epilepsy, a first invasive examination is done, and the patients are implanted with electrodes in various different areas here. So each of these points here is one electrode contact in order to be really sure where the epilepsy are coming from. Then the patient is waiting for seizures to occur for a period of 1 or 2 weeks. And then if all goes well, the neurologist or neurosurgeon can identify where the origin of the seizure is and resect this area. During this period now, when the patients are waiting for the seizures to occur, cognitive neuroscientists can knock on their door and ask them whether they are willing to participate in, for example, this paradigm here. And so in this study here, what we looked at was, again, the representation of individual events here while they were being studied. And we focused on 2 main areas. So on the one hand, the lateral temporal cortex that is relevant for the semantic item representations itself. And on the other hand, the dorsolateral prefrontal cortex, as mentioned before, the source of inhibition in this paradigm. So does this effect here rely on erasure, inhibition, transformation, or selection? So if it relied on erasure or inhibition, one would assume that the representation of individual words, while they are initially shown, and while they are afterwards shown during the test, so at the end, we show all the words again and ask the patient whether they've seen a particular word or not, one would assume that the representations of words when they are supposed to be forgotten during the initial presentation and during the final presentation is very different, right? So they see a word, they are asked to forget it, they suppress the memory trace of this word. If they see it afterwards again, then this should be represented very differently from the original experience. So basically, the similarity between the encoding and the retrieval event should be much higher for the to be remembered words than for the to be forgotten words. One could also assume that the attempt to forget a particular word is due to memory transformation. So again, one would assume that there's a low similarity between encoding of a word and final retrieval of a word because the memory trace has been modified due to the forgetting instruction. Now, one could also assume that it's maybe the representational format that is changing due to the forgetting attempt. So that then one would assume that maybe there's a particular aspect of a memory trace of a word that is similar for the remembered words between encoding and retrieval. And there's another aspect that is similar for the words between encoding and retrieval when the occurrence of this word has been suppressed. So now what we tested here is the amount of similarity between encoding and retrieval of individual words. And first, for the to be remembered words, we found that during the retrieval, there was a selective reoccurrence of an item-specific memory trace in the lateral temporal cortex for specific items in the gamma frequency band that has been associated with activation of a brain area. So when you see a word, there's a particular pattern of gamma activity. And when you afterwards retrieve the same word, the same pattern occurs again. For the to be forgotten word, this is not shown, but what we found is that when we see a particular word and the memory of this word is suppressed, then when we afterwards show the same word again, we still see a reoccurrence of the original pattern, but now not in the gamma frequency range, but in the alpha frequency range, which is associated with inhibition. So basically, it's an inhibitory part of the original engram that is reoccurring, remains in the memory trace for events, even if they are afterwards forgotten. We did not only find this reoccurrence of this inhibitory pattern of forgotten items in the lateral temporal cortex, but also in the pattern of activity between lateral temporal cortex and the prefrontal cortex. So again, there was a reoccurrence of these inhibitory patterns when we showed a word that the participant was successfully able to delete from memory. Okay, so to summarize, I started by showing in the first study that stressful memories are remembered in a particular way, namely they are remembered by a generalized pattern of activity in the amygdala. For traumatic memories, we found that there were two particular aspects of the representation of formats. On the one hand, there was again an overgeneralized representation at a sensory level, but then a very distinct representation of the individual traumatic movies on a conceptual or semantic level. We found some evidence that self-incongruence, so shameful memories, affected the representation of formats of memories at the phenomenological and the semantic levels. So at the phenomenological level, because they were remembered more often from a third-person perspective. At the semantic level, because they were more similar to other shame-related memories. And finally, we found that voluntary forgetting affected representation of formats at the level of neural oscillation. So there's some evidence that even after successful memory suppression, a memory trace remains, but it's only the inhibitory part of the engram that stays there, whereas for successfully encoded items that are afterwards remembered, it's excitatory aspects of a memory trace that can be found. So this is basically the gist of these initial individual findings. So I would like to suggest that trauma, stress, and also memory suppression do not only affect what we remember from our past, but also how we remember it. And this can be studied with cognitive neuroscience. So I would like to thank you for your attention. I would like to thank all the team members here during COVID and afterwards, and some of our funders, and I'm happy to take questions.
Yes, thank you for your very explanatory lecture. And yes, if somebody has questions or comments, please. Thank you very much. Very, very rich lecture. So a lot of questions on the way, but just maybe one broad, but not question, or maybe you could comment something on traumatic experiences which are not reachable by our verbal memory. Yes, I mean very early experiences. Do you have any suggestions and how they can be transformed or changed, or like something like integrated?
I don't really have a theory about that. So I think what these studies here are in a way of like an experimental preclinical model of is individual, also not recurring, but individual traumatic memories during adult life, right? So inducing post-traumatic stress disorder. So it's known, as you mentioned, that traumatic events that occur during childhood have a maybe even more severe effect in many cases. but in many cases, as far as I know, they also do not induce typical post-traumatic stress disorder, but in particular other kinds of diseases. They may lead to personality disorders. They, in very severe cases, may induce symptoms of autism and so on. So I think these studies that we did and the phenomena that we investigated may capture some of the aspects of intrusive memories of these later life traumatic events. I don't think they are really an appropriate measure of early life trauma. So I don't think that's maybe a satisfying response. Yeah, I mean, I think it's probably again some like phenomena that can be explained by specific representational formats. So these representational formats do not need to correspond to voluntarily accessible and sensory aspects of an event. They, for example, they could also correspond to bodily feelings. So there's like the phenomena of body memory. So involuntary somatic feelings of pain, for example, that reoccur, that are not consciously connectable to the traumatic events, maybe in some cases. Yeah, and there can be other psychiatric diseases, like, for example, the ones that I mentioned, but also others that are induced by these early life traumatic events that are very different from these features of intrusive memories in adult life.
Some other questions?
Thank you for a very impressive work, and I think it's a very special occasion to have your lecture here at our university. Yes, I'm associate professor of psychotraumatology here, and we do a lot of research in traumatic stress. From your presentation, I understood that our brains has a tendency to overgeneralize trauma triggers. But so it seems kind of normal. Yes, it seems. So what's behind it? Maybe you have an explanation why our brain react this way. Thank you.
Okay, yeah, thanks. So, I mean, obviously the brain may change as like both in its function and its structure due to traumatic events, but these changes, I believe, can to a certain degree explained by the normal functioning of the brain, and in particular the normal functioning of the amygdala. So so what I would think is that like one of the functions of the hippocampus is to tease apart memories in various different ways. So this has also been referred as pattern separation, right? So to make sure that when you see a particular person, you can differentiate this person from a similar person, right? Even though they may share some perceptual or conceptual or whatever features. Another function of the hippocampus is to disentangle events based on their different contexts. So something that happened at one place in time can be disentangled from something that happened at another place in time, and in particular can be disentangled from something that is happening now. And the amygdala is in a way doing the opposite. So the amygdala is generalizing aspects. So this is something that you can appreciate during conditioning, right? So the effective valence of the unconditioned stimulus is spreading over from the unconditioned stimulus is spreading over to the conditioned stimulus. It may also affect the entire context in which something is occurring. And this is of course beneficial, right? Because events that are temporally, spatially, semantically, perceptually associated with a very salient aversive event should also be avoided, right? So I want to add this aversive label, if you want, to all of these events. So it's beneficial, it's adaptive in many situations in life that there's this generalizing, spreading function of the amygdala. So now what happens in what happens in traumatic events is that this is exacerbated at the cost of the separation by the hippocampus. So basically, something that occurred in one context, the traumatic context in the past, is spreading over and is affecting what is happening now. So even though a person is not in the same place where a traumatic event occurred, is not seeing the same people, is maybe in a completely different life context, is still cannot differentiate the original situation. And this is also why many trauma therapies, as far as I know, aim to make the patient realize that there's a difference, right? So there's like the narrative exposure theory, for example, where the patient is made to realize that the traumatic events is not happening in the here and now, but it's happening somewhere else and at a different time. So, but I think that like these both processes, like both adaptive processes and this psychopathological symptoms can be explained by the same basic mechanisms of a set of brain areas, so in particular amygdala and hippocampus, of course, in interaction with other areas like ventromedial prefrontal cortex and so on, but they are probably those that control most of these processes.
Are here any more questions, or maybe those who are in Zoom, you have a possibility to ask questions in chat? Yes, please.
Okay, thank you for presentation. It brought a lot of thoughts, but just some futurologist prognosis. So do you believe that time will come when you will, so neuropsychology or neuroscience will be able to, let's say, to diagnose entirely on the basis obtained from the functioning of the brain, not just from behavioral observation, not from subjective reports, but just enough data will be enough just to say what is happening.
Yeah, that's in a way a very interesting philosophical question. Well, I mean, as you know, this has been the goal for DSM-5 and even DSM-4, and yet currently no single disease in the DSM uses brain imaging for diagnosis, and this may have many reasons. Yeah, so in particular, probably the incomplete mapping of diagnostic ontologies onto biologic individual brain systems. So it's not that depression is like one unitary disease that can be mapped on one brain system, which is like more or less perturbed. I think that this is probably one of the main problems. Then, of course, still the lack of sensitivity, but you can always say, well, we need not only 3 Tesla, but we need 7 Tesla, 9 Tesla, and more powerful computers. I think actually the problem is deeper. It's more of a conceptual problem. Nevertheless, I think that hopefully cognitive neuroscience can contribute to an understanding of psychopathologies and also to an understanding of links between symptoms that may not be obvious from a purely behavioral or symptomatological point of view. So one may see that different processes, like for example, this overgeneralization or lack of separation, can be seen in various different diseases. So it's basically the idea of overlapping mechanisms and research domain criteria that I think can be supported by neuroimaging and general cognitive neuroscience methods. So I mean, the short answer is I think I hope there's a place and there's a relevance of this kinds of methods. I don't think that they can ever replace diagnosis. Can they support diagnosis at some point? I'm not sure. I I mean, there's some diseases like Alzheimer's disease, right, which is like sometimes treated as a psychiatric disease, sometimes as a neurological disease, depending on where you look like. I think that's different, right? I think there, brain imaging, structural MRI, maybe even functional MRI, PET, of course, maybe EEG can very much contribute to the diagnosis. It's much more difficult in classical psychiatric diseases like depression, for example.
Thank you very much for a very interesting presentation. I am curious regarding amygdala and hippocampus. Like, we know that both of these structures have many receptors, receptors for glucocorticoids, mineralocorticoids, even sex hormones. And during stress, there are many substances that are released by the body. So can we have any predictions, for example, if person's stress response is that he released a lot of cortisol or maybe less cortisol, can that or any other substance, can that anyhow predict this intrusive thoughts or any trauma appearance?
Yeah, thank you. So, I mean, this is not my main area of research, but I would guess so, right? I mean, like some of the differential responses of amygdala and hippocampus to stressful and like moderately stressful, but also traumatically stressful situations have been related to the different composition of receptors of mineralocorticoid and glucocorticoid receptors in these two substances. So I mean, my sort of like half level interpretation is that you are now the inverted U-shape relationship between cognitive functioning, and cortisol concentration, right? So there's an optimal functioning at medium levels of cortisol, and at both very low levels and both very high levels, the functioning is reduced. I think this is supported also by like receptor studies. The peak is differs between different brain regions. So the prefrontal cortex, for example, is extremely sensitive to even at like moderately high levels of cortisol, its function is reduced. So this is why in exams it's so difficult to find the right retrieval cues, and the prefrontal cortex working memory is not well working. The hippocampus is a little bit more robust. So stressful situations, like moderately stressful situations, like exam situations, are relatively well encoded, right? So this also makes sense because these are situations that are often relevant for us, right? So these are the relatively unique events we need the episodic memory system for. The amygdala, on the other hand, is even more robust and can be even activated in extremely stressful situation when the cortisol concentration even exceeds the optimal regime for the hippocampus. And this is why in extremely stressful situations, the amygdala functions, like the generalization function of the amygdala, is still existing. It's actually even prevailing, whereas the narrative binding function of the hippocampus, but also the function of the hippocampus to pattern separate and to represent distinctive context is already reduced. So there's, I think overall there's already quite a bit known about the overall macroscopic density of these different receptor types. If you look at the details, so in which layers of the hippocampus and which subregions of the hippocampus which receptors occur, and in particular how the situation is in humans, and how what the degree of individual variance is, and whether this explains the resilience to stress or trauma. I think there's still much to be found out, but I think that's the right questions to ask.
Some other questions or comments? Some other questions? No, yes.
Maybe just to follow up on the previous question. So what can we do to make us more robust? Any advice?
I'm not sure whether my expertise as a cognitive neuroscience researcher is sufficient there. Well, I guess of course, we want to avoid traumatic situations, right? That's like in the first place. Once they've occurred, there's certain, well, I mean, I think also after like moderately stressful events, there's like relatively beneficial coping mechanisms like reappraisal, reframing that can help deal with that. But maybe your question was more, how can we like increase the resilience to make sure that after like a moderately distressing events, we are not, well, do you have any ideas?
Especially with the fear, if you are fearing of and you are experiencing it every day, every hour, and at the end of the day, you will be less fear.
Yeah, thanks. Yeah, I think also that's like one idea of exposure therapy, right? So you experience like the view of spiders, you find out that they're not dangerous. I mean, I think in many cases, at least in therapies, that's relatively well working. Sometimes it doesn't generalize to other contexts then. So if you come back to your basement and you see the spider, and maybe you're still nervous. On the other hand, it's this may not be a strategy to increase overall our resilience to all kinds of distressing events in life, right? I mean, I think there's like from an epidemiological perspective, right? There's many factors that can increase resilience. So, for example, like a psychosocial, like a healthy psychosocial network. I guess I don't know if your overall lead a relatively rich and fulfilled life, I think you're typically more resilient to also distressing events, but maybe that's a bit trivial. Also, I mean, that's also something that another question, how can you make your children more resilient to potential distressing events, right? And, I mean, it's hard to run any studies on that, like prospective studies. You can like try to find out some factors. I'm not sure whether I'm the right person there to give you any scientific responses.
Some more questions?
Hello, is it possible to take a question from behind the screen?
Yes, please.
Great, yes. I was really very fascinated about the lecture. My question is about research that has been done by National Institute for Clinical Application of Behavioral Medicine, and about brain research putting together with different trauma therapies is one of the real great ways. And now my question is that trauma therapists have shown that when people change the meaning of the traumatic events, they at the same time, they pick up different memory traces from the same event. And when this meaning changes, then they start to believe those new memory traces, they have not been picked up before. So my question is how your experience could explain this clinical experience, because it seems to me that flexibility in brain is such a huge possibility for us, that when the person changes the meaning of the traumatic event, then also the memory traces will be changed.
Yeah, thank you. Thank you. That's a great question. I mean, in a way, it's more like a suggestion or a comment if I get. So maybe the question could be how to reconcile that with the like some of the findings that I presented and like the theoretical ideas. And what I would suggest is that if you change the meaning of an event, either by yourself or through some therapeutic intervention, this elicits this modifies or makes more accessible actually some conceptual or narrative level of representation of the same event. So I would argue that at least in many cases, the memory trace of the original event is not modified or changed, but is a specific aspect of that memory that is more strongly activated. So basically an interpretation of the original event that in principle would have been possible already from the beginning, but for many reasons. So for example, because the conceptual, semantic, contextual interpretation of the event and by the hippocampus was reduced, was not accessible. Now this new type of conceptual framing and interpretation is accessible. So basically it's again, to formulate it differently, it's a different kind of schema that is now made more accessible, that is already right from the beginning a potential part of the memory trace. So I would fully agree that like these interpretations are, I mean, that's in a way the classical way how psychotherapies work, or many psychotherapies at least, that they modify the meaning of what has occurred. They don't modify the specific contents that are being remembered, but how they are being remembered. And I would think that this is like a high conceptual, semantic, narrative level that is modified, for example, also such that it can be better linked to other memories, and that it can be better linked to the view of myself. So that these memories are less associated with feelings of self-incongruence and are in a way accepted as a part of one's identity.
Thank you so much. It was great explanation. Thank you very much.
Thanks.
Okay, one more question, or can we finish now? Okay, if there are no questions in the moment anymore, so I would like to thank Professor Nikolai Axmacher for a very interesting lecture and for openness to the questions. So thank you very much.
Thank you. Thanks.
Thank you very much. And I would also like to thank all of you being here today and for having your attention. And everybody who has applied for a certificate can come to me. We have prepared them for you for participation. And yes, for those who are participating in person, I invite you to continue discussions in more informal way outside the room. For those in Zoom, thank you very much for your attention and have a great evening.
Thank you.